Welcome to the tour of the animal cell. The difference between an animal cell and a plant cell is mainly that an animal cell has centrioles and lysosomes, which plant cells do not contain, and it does not have chloroplasts or a cell wall. Cell membranes are arguably the most important organelle of the cell. Membranes control what comes into and what goes out of a cell. Membranes are composed of a phospholipid bilayer that forms the strong boundary between the cell and the outside environment and gives the cell its flexible structure. The bilayer is made up of hydrophilic heads on the outside and hydrophobic tails on the inside in order to create an effective boundary. In all cells, proteins can be found embedded in this bilayer. Peripheral proteins reside on the outer sides of the membrane, and their function is determined by their location. Integral proteins can also be found in the bilayer, running all the way through the membrane, acting as transport proteins to assist molecules across the membrane. Carbohydrate chains can also be found on the outer portion of the membrane so that other cells may identify the cell accordingly. Passive transport is the diffusion of molecules down a concentration gradient without the use of energy from ATP or another source. Passive transport can either be small uncharged molecules moving through a cell's semi-permeable membrane, or larger charged molecules moving with the gradient through their transport protein located in the cell's membrane. Diffusion and osmosis are both examples of passive transport. Diffusion is the process by which molecules move from an area of high concentration to areas of lower concentration in an effort to be at equilibrium with surrounding molecules to create an isotonic situation. An example of diffusion is that if you add salt to water, the mass of salt will attempt to divide itself evenly along the water molecules in order to become evenly divided. Osmosis is the movement of water from a higher concentration gradient to a lower concentration gradient, again in an effort to be at equilibrium with surrounding molecules. An example of osmosis is water experiencing osmosis into a cell in order to stabilize that cell with the rest of the environment. However, if there are not enough cells in the solution, the cell will be hypotonic to the solution and inevitably bust. Active transport utilizes energy from the cell, usually in the form of ATP, in order to move nutrients into or out of the cell according to the against the concentration gradient in order to maintain homeostasis. Usually active transport occurs in the form of endocytosis, or the moving of nutrients into a cell, or exocytosis, the process of removing waste from a cell. Some more complex forms of endocytosis include phagocytosis and pinocytosis. Phagocytosis is the process by which a cell eats. In phagocytosis, a cell will essentially engulf bacterium or some sort of molecule in order to process that which was consumed and convert this food into energy. Pinocytosis is the process by which a cell takes in water against the concentration gradient. The main difference between active transport and passive transport is the fact that active transport utilizes energy and moves against the concentration gradient, and that passive transport does not require energy and moves with the concentration gradient. Both passive and active transport utilize transport proteins. Active transport uses protein pumps to move molecules either in or out of the cell. Both are incredibly crucial to the cell's well-being. Coming into the nucleus, we must first enter through the nuclear envelope. The envelope is perforated with pores that allow transport in, through, and out of the nucleus. The nucleus is the most conspicuous organelle of the cell and contains the hereditary information and DNA of the cell. It is also acts as a brain and a control center for most of the cell's processes, including protein production. The nucleus is site chromatin. Next, we're going to be talking about chromatin, which expand during cell reproduction in order to form chromosomes. And these, the chromatin normally reside inside of the nucleus in just a big old soupy structure that looks like, spaghetti noodles all coiled up together, and you can't really tell which one's which. And it's like it would take forever to unwind those things, and that's how you know when you're looking at chromatin. Chromatin are important because without them, the cell would not do so well during cell division. Centrioles are also very important in cell separation. This is a centriole, as you can see. Centrioles assist the cell in helping to keep its shape during reproduction and also keep the chromosomes dividing correctly. This is the cell's cytoplasm. In the cytoplasm, the cell houses every single organelle and the cytosol in which they reside. much of which we will be traveling through today, is cytoplasm. Thank you. This is a three-dimensional representation of the smooth and rough ER. First we will be talking about the rough ER. Located on the outside of the nuclear envelope, the rough ER gets its name from all of the ribosomes littered across its surface. These ribosomes attach and help in protein production. The rough ER synthesizes proteins that are made and that are used in the cell membrane. The smooth ER lacks ribosomes on its surface and contains many enzymes that perform specialized tasks, such as synthesizing lipids. After proteins are produced in the ER, they can move to a stack of compressed membranes known as the Golgi apparatus. The Golgi apparatus attaches carbohydrates to lipids and proteins and then ships them to their final destinations, much like FedEx. The flagellum helps the cell to move by snapping back and forward in a whip-like motion. Located on the outside of the cell membrane, this is one of the most important things in cells in terms of movement. Males have no flagellum in their body, except for sperm, which lose their flagellum when they join with the egg. Lysosomes are basically the stomachs of cells, and they break down lipids, carbs, and proteins into particles that can then be utilized by the whole cell. Lysosomes also work to break down organelles that are no longer useful. Both microtubules and microfilaments assist in making the cytoskeleton, which helps the cell keep its shape. However, the difference between microtubules and microfilaments are that microtubules are larger than the microfilaments, and they also are in more tubular formations, while the microfilaments are more in a strandy formation, and they're also much smaller. Mitochondria utilize the energy gained from food to create great energy compounds in the form of ATP that the cell utilizes to power growth, movement, and development. This is one of the most important organelles in the cell. Located in the nucleus as well, the mysterious nucleolus. For years scientists puzzled over its function, but we now know that the nucleolus begins the synthesis of ribosomes. Let's take a look at ribosomes. Ribosomes produce protein, and protein executes most cell function. Ribosomes are composed of RNA and protein and can be found on the rough endoplasmic reticulum, are floating freely in the cells. This is a picture of a vacuole in a plant cell. They are much larger than the ones in animal cells. In animal cells, vacuoles work as storage areas for the cell. They're basically like the warehouse of the cell. They store proteins, carbs, water, and salt for later use. There's also a smaller type of vacuole called vesicles. As I said before, vesicles are smaller versions of vacuoles. However, vesicles act like, in addition to acting like warehouses, vesicles are usually involved in transporting substances intracellularly. Coming to a close of our tour, the cell theory states that the cell is the fundamental unit of structure and function in living things, and that all living things are composed of cells. 3. That cells come from pre-existing cells. That concludes our tour through the cell. Bye.